at the Baylor College of Medicine, Houston, Texas, who's going to tell us, what was the title, Lynn? I don't know. There it is. Thank you very much. Oh, it's a long title. A new non-viral, non-toxic DNA minivector for basic and clinical research. How basic DNA topology research opened doors for gene therapy.
There you go.
Lynn. I should say also that Lynn has, as DeWitt mentioned, started up a company with a wonderful name, Twister Biotech. She may tell us a little bit about that, but there are leaflets which you can pick up outside after the lecture. Perhaps we'll have questions after the 3 talks, just before tea.
All right. Well, thank you very much, and thank you for giving all the introduction. So, in fact, I got into this field because of the very, very rich and amazing and beautiful mathematics behind it. And I've been fascinated with it, well, I won't say how many years. But anyway, oh, you said I have to use arrows. There they are working. So I think I don't really need to go over a few definitions, but I do this to the biologists, so I force them to learn that link LK equals twist plus drive. And that's a very important convention, but really let's go right on down to here. For me, when I talk about underwound DNA, by convention I mean negative supercoiling. I'm going to go with the biologist language. And when I say overwound, that's positive supercoiling. We are going to talk a little bit about that. So what about DNA in cells? And I needed to go back just a step because talking to a few of the mathematicians around here, I realized, okay, I think that we're going to back up just a bit and sort of bring us up to speed. So DNA in cells is not, as it's pictured so beautifully by the crystal structures. In fact, in cells, it is rarely in this form. In fact, biologists would tell you that this is dead DNA, completely inert, inactive DNA, because in real DNA there is underwinding. Remember I told you about that's the negative supercoiling. There's overwinding and transient extremes of both of these. Sometimes this winding is actually fixed so that writhe is precluded, somewhat like Dorothy's little pictures there, and sometimes it's not. There are never ends, and this is a really important concept because I keep getting people saying, oh, but you know, human DNA, there's ends. Those ends are extremely fixed. There's telomeres, there's proteins, they're embedded. So for all real purposes, although that's linear DNA, it can be treated as if it's closed circular. So the topology for bacterial DNA is the same as for human DNA. And not only that, but of course there are electrostatics, there's counterions, there's packing interactions, not only of DNA with DNA, but DNA with proteins and DNA with membranes. And there are many and multiple, sometimes extreme forces on the DNA. And so Dorothy, pulled one of these so tightly, in fact the memory is still going, there's a little bit of stretch here. And some of the forces that are put on DNA in a cell can exceed the force so that it can't stretch back to its normal normal original shape. So that's actually been something that's fascinated me for a really long time. And so I just wanted to share really one of my favorite curves ever. This is where most of us work in the in the lab, in test tubes, or in a lot of physics and a lot of single molecule stretching experiments, and that is with linear DNA. And so that, of course, is, that there's actually a double helix underneath that. So this is that little drawing here because it's kind of hard for me to draw, although Dorothy, I saw you did a beautiful job. So at best, people will work with circles, but they're relaxed or they're linear. And that is dead DNA, as I've already told you. And so what you have to do is introduce what we biologists call negative supercoiling before you get, I think, a very interesting transition, a very tight inflection. And I've been fascinated by this inflection point where biological activity occurs. This curve is actually shown for the site-specific recombination event that DeWitt was talking about. But in fact, it's true for recombination, it's true for replication initiation, it's true in yeast, it's even true in human cells. So this basic work that we started with E. coli has held true across the kingdom, the domains of life. So this supercoiling homeostasis, this perfect balance where twist and writhe are maintained and really all cells examined, is something I'm very, very fascinated by. And that's all I'm going to say by that, but that is the motivation behind my work. And so I will give a shameless plug for a quarterly review of biophysics review that DeWitt and I and a few others have written. And we talk about what could be the explanation for this as you increase negative supercoiling, why you get an increase in biological activity. And I'm going to have a hard time, let's see, put this down here. What I'm shown by the increase in negative supercoiling is I'm underwinding this, and what happens is that DNA coils up, right? So that is what's depicted along this axis. And so it's not accidental, I think, that cancer cells, the DNA in cancer cells, or in rapidly growing microbes that have infected your body, the DNA is living about here, hyperactive. So I think it's really important to have this concept of threshold, because biologically you want to shut it down when you need to, and have it active when you need to. So it's really a great place to maintain DNA, and that's in that review if you're interested in that. So here's a little more. I love your simple drawings to it, but this is what DNA really looks like, underneath here, and then we just draw it as a curve. So here's a really big plasmid. And what we wanted to do is we wanted to understand, how does DNA really behave? Is it really this perfect elastic medium? We already knew it wasn't, because in force extension stretching, sometimes you could stretch it so far it wouldn't come back. And the forces, as I told you, of polymerases naturally in cells can exceed that force. So I said, okay, this is not the perfect elastic medium. So to reduce all these other variables and get rid of all that other stuff that really is thermodynamically not defined anyway, I wanted to get the tiniest little circles that I could get, because there's been some, again, wonderful mathematics and beautiful physics done with these sorts of hypothetical circles, but I wanted to start testing them. And I'm not the first to have this idea. In the 80s, people were already doing this, and so it's just that we wanted a lot of these mini circles because we wanted to do a lot of experiments. And all the methods previously were not, in fact, that's a point I make here. If you try to take linear DNA and bend it around and ligate it, you have to get the right kind of bending sequences. And of course, the persistence length is a bit of a challenge. So you can get some circles, but with very, very poor yield. So I really wanted to use this site-specific recombination. Fortunately, I joined the Cazarelli lab. They had already worked out how to do it, so all I had to do was try to figure out how to get this little bitty circle, because in fact it had never been seen on the gels, and that's why these products in the old days were referred to as deletion products. So you started with something, you lost some DNA, and you recovered the deletion product. Well, so we went after this missing little bit, and we really did it to understand this curve, as I've already shown you. Here are some atomic force micrographs of a 330 base pair DNA. Here it is when it's relaxed. Here it is with a little bit of one linking number change in the negative direction, so on, until it's basically a tiny little crazy rod. So when I first wrote a grant on this back in the US, they said, this is crazy, this is not biologically relevant, it will not be active, you've made something that is irrelevant. And so I thought, well, that's really interesting, because if we treat this thing with a human topoisomerase, in this case topo2, it relaxed it immediately. If we treated it with RNA polymerase, another enzyme, it would transcribe it immediately. And I said, well, it's not quite an inactive freak, because these things are working. And that led to the biological hypothesis that these tiny circular DNAs of a few hundred base pairs might be the vector to use to get into cells, because that's a really big challenge. And we don't really have time to go into all the challenges, but most of them have to do with the same reasons that you couldn't get a lot of information by using really big, undefined plasmids to try to measure behaviors of DNA on the length scales that I'm interested in. So these are really big, and these are really small. And it just seemed logical to me that they would get into cells. And indeed, these guys, these mini circles, they get, the word is a medical word, transfect. Maybe it's just a molecular biology word or a cell biology word. I am not that. But they transfect and get into every cell type we have tried. They express genes, they express shRNAs, and they express microRNAs. And some of this is published. So that was very exciting, and that's already a product. and we're going to get in a little more into the company that we have started, but I just wanted to take you through one more bit of data, and that is, how do you get, so cell culture is one thing, but if we really dream of getting into humans and trying to do something toward human health, the applicability of direct basic DNA topology analysis, we have to think how to get it into a person. Well, one way is to nebulize it in a nebulizer such as this, and it makes this really fine mist that the patient can breathe breathe very deeply. And I think this is standardized across many countries. I know Europe uses it, Canada uses it, Australia. I think even in Asia they're using the exact same type of nebulizer for lung treatments. So the reason you can't use gene therapy and deliver it that way is that the minute you put DNA into this little reservoir, the forces are so high that the DNA is immediately sheared. So that was a hopeless, you can't even do it. And so ours are really small, maybe we could try it. And indeed, it works. But let me show you first. And I actually drew these to scale because I think it's very, very striking, the difference of the typical plasmid. This is P Super. It's something that is selling a lot. People are buying this to put into cell cultures. But if you put that in that little nebulizer that I showed you, it is almost immediately broken down into tiny, tiny little linear fragments of DNA. So not only do you not deliver your gene therapy, but in fact you deliver what is potentially a toxic byproduct of DNA. So this can induce DNA repair, inflammation, apoptosis, and in fact we now know that it wasn't, the patients didn't die because they didn't get the therapy back in other people's trials, not us. It was because you were introducing this toxic breakdown product of DNA. So here's what the other plasmids were. This is why they abandoned as a hopeless point. And we just said, how small can you go before the thing will actually survive the shear forces that are in nebulization? And you can see these are the gels. So here's the plasmid going away. Here's a 2000 base pair plasmid, and it survives pretty well. You can see an accumulation of these same sized products. It's something about the radius of gyration, about the shear forces of the nebulizer. So I think that's very interesting. And here's a 1200 one. Very, very little degradation. Of course, the littlest guy we tried was not nebulized at all. So this reintroduced the possibility of using a nebulization to introduce this into mammals. And if you plot this, I love the shapes of these curves. I was a math major for a little while. I love to look at how things look. But this is the very rapid, almost pseudo first order kinetic decay curve for the typical use plasmids, and you see they're completely worthless. And as you start getting into this interesting area, they'll be a little bit, they can survive a little bit, and then they decay. And that's explained because as the reservoir volume goes down, the cycling is faster, and then the DNA is shredded. And so this can be, we can actually increase the length of the DNA that we can put into mammals by simply stopping the treatment after 5 minutes, opening the lid, and putting more liquid in. So you keep that going a little more slowly. But we can't rescue these plasmids, but we can get this out to about 2,000 base pairs. So anyway, these concentrate because the water will evaporate eventually through the treatment, and so by the end of the treatment, they're actually, the DNA is completely survived and completely gets in. So let's go straight into mice, and this is the brand new data. Some of my friends have heard me before, so when I came to give this talk last week, I said I can't show the same old data, so I was very excited that we had new data to show. And so using that same nebulizer, we put either minivectors into mouse or just the water, but aerosolized it and had the mouse do the same treatment. So that's the control mouse. And I don't know, have any of you ever done a mouse experiment in this room? You know, not our thing, right? So a big steep learning curve for me. Anyway, but we knew that our little minivectors encoded this tiny gene that was not secreted. Now this is important because it would stay in the lungs and not go out of the lungs. And this was a luciferase. So when you give it its substrate, it will now fluoresce. And the mice were given the minivector either mixed with water, which is a dream because you're not introducing any weird chemical into the delicate lungs, or with polyethyleneamine. And after some time, you give the substrate for this luciferase gene, and you just ask, was there any luciferase activity? Because if there were, it had to come from the gene therapy into the mouse. And so what you're looking at is the first successful gene therapy through the lungs into a mammal. And so the water didn't work as well as the PEI. We didn't expect it to. But there are so many instances in medicine where you don't need huge expression of a gene. You just need a little constant expression of a gene. So we're very excited that we might be able to even just use water to deliver via nebulization into mammals. So that's the mouse data. But now what's another way to get into the human body? And of course, that's through an intravenous delivery. And I've just grabbed this off the internet. Maybe some of you have had the misfortune of having to have intravenous drug delivery. The problem, and this is what we all learned, whoever took a biology class, is that the blood is filled with nucleases. And the nucleases will immediately destroy DNA. And so there's no hope in ever delivering gene therapy through the blood. So we just said, well, let's just see if these little guys, why would they? But good thing we tried this crazy experiment, because here's the typical size plasmid, and it's just incubated in human serum, and you see that it's digested away. Now this is now a bunch of nucleases that reside in the human serum. And here is synthetic siRNA, which is just small inhibitory RNA, or short inhibitory RNA. And you can see those are also very, very rapidly degrading, and there must be 900 companies across the world that are started on various ideas trying to protect or block or use or hide or fool the blood into not recognizing this, because it would work great as a gene therapy delivery if you could only get it to survive the human serum. But this is showing how it fails. And if you plot these, they just fail very, very rapidly. But in fact, the minivector, and this was a shock, and we've repeated it a million times, it's now published. Please go download this for free, because it's free to you, but it cost me a lot. And I did it because I get emails from Poland, from all over the place saying, I can't get your journal, please will you send it? So I paid to have these things free, so please feel free to download them. So anyway, they completely survived. I mean, there was no degradation. And I learned, so I am trained as a protein enzymologist, that DNase, or serum nucleases, just hit DNA. So the question is, why in the world are they not hitting these small DNAs? And that's still an open question that we're probing right now. So it's fascinating, but the truth was they survived. So you can tell we're really getting excited at this point. So, in summary for the science part, minivector DNA survives both mechanical shear forces, and there was a lot of really neat physics behind the development of that nebulizer. I was really fascinated by that. It also survives, and it transfects every cell type we have tried. And if you deliver the minivector DNA via aerosolization into a mouse lung, we got a functional gene encoding a functional protein out. So now, of course, we're putting in real disease, important disease genes. But this was just the proof of principle, because it's a lot easier to assay. The valuable postdoc years, as DeWitt referred to. So DNA survives the degradation by the serum nucleases, which is still an absolute shock, but the truth was it does. And a really important point is that the topology matters. I didn't show you all the topology experiments here, but in fact, twist has to be avoided. So we have to manipulate our circles to minimize twist if we're going to go into the intravenous delivery. And at the same time, writhe is absolutely required for the nebulization survival. So who would have thought of that? So I'm almost laughing, and now I'm one of them, but all these companies about, I'm making a vector, I'm improving this vector, I'm going to nanoconjugate that. Again, hundreds and hundreds of companies have opened and folded because they were going to try to circumvent having to really understand about twist and writhe. So it really comes down to this. So at worst, what we've already shown is that minivectors provide a new tool for basic and clinical research, because we can do things in mice and we can do things in the cell culture. At best, this may represent a major breakthrough for the bottleneck of gene therapy delivery, which is really the only problem. We can cure so many diseases if we could just get the DNA to the cells you need to get them to. So DNA topology and the mathematics and the physics underpinning this field has really led to this new tool that has been, we're just having a lot of fun with this new tool. And I think this bottom point is one that I am stressing all over the States because we're becoming more and more and more driven by translational research. I did not set out to make a gene therapy vector. I set out to answer a very basic question in DNA topology field. And in answering that question, we've suddenly got a gene therapy vector. So one never knows when these sorts of leaps or jumps or hops might happen, so we have to keep funding basic research. It's what I really, really feel. So I just wanted to tell you a little bit about our company. We did start the company. We call it Twister Biotech. It's live, it's online. And I wanted to tell you a little bit about our commercial partnering opportunities going on right now. And you already know what the minivectors are. We can make milligrams and milligrams of them. So they're a non-viral DNA vector with bacterial sequences. I didn't really mention this, but there's no bacterial sequences left. And we've also stripped out these things called CPG motifs. Those are silenced by methylation. So if you get rid of all of them, and we can because it's so small, then you're not going to be silenced and you can express the gene longer in gene therapy. So this is a very important point that no other gene therapy vector offers. And it's very important in Europe because the European regulatory agencies are really moving toward an insistence of not introducing bacterial sequences or antibiotic resistance in the gene therapy arena. So right now, ours is the only vector that fits that bill. And so the two arms of the platform are for basic biomedical research, which is what I've already shown you. And these are some of the things I've already talked about, but I didn't mention that there's no inflammatory response. Really, the pathology, the histology of the lungs and the mice when we've delivered these things, it's remarkable. We haven't carried them out for years. There's a lot more toxicology to be done, but there's just really zero problems with having these vectors. Unlike the plasmids, which you have all kinds of inflammation, all kinds of apoptosis. None of that with our vectors, which is, again, really fun. But there's also the therapeutic platform. So we're calling these next-gen gene therapy products, and we're using them for DNA vaccines, et cetera. And so the therapeutic and the biomedical research are the two arenas that we're selling. So we've outfitted the laboratory, and there it is. And there's our team in the good manufacturing practices. We're ready for GMP production. We've built the website, and this is what I showed at the conference last week that I guess apparently led to this invitation today, is this is our website. And I said this because I had so many friends in the audience who were always saying, Lynn, can you send me something? Lynn, can you send me something? And what I found is, why aren't we accomplishing our goals, right? And it's because everyone in my lab, including me, are making them for everyone else. And that's the perfect recipe to start a company. So that's exactly why I did it, is to offload the burden, get back to what we thought were really, all of those are great experiments that everybody wants to do with them, but we want to do our own experiments. So you can build your own, you can order your own. Please order them, please buy them. I'm not making them anymore. I don't even do it in the lab anymore. They're so much better than I am at making them in the company. We have a patent portfolio. We have one issued patent, we have one about to come out, and we've just filed 2 new provisionals just this summer. We've got 2 more going out really, really shortly. And of course, Twister is attending vendor shows, and here they are doing it. And I want to focus, if you cut out the people and enlarge that a little bit, they are even carrying the DNA molecules, these sorts of props, to these vendor shows, because it really boils down to, look, what you have to understand is that linking number equals twist plus rive, because that's what it boils down to every single time. And you may want to, if there's business people in the audience, Bio Europe is just a 50-pound train ride away from London. So you can go see Twister, they're going to be there. And let's see, we are getting orders. But it was a very unsatisfactory. I wanted to frame our first US dollar from the order, but it was a PayPal. How do you frame a PayPal order, right? It was very, not like the old days. And of course, we're very actively pursuing the commercial partnering opportunities. And I'm going to go a little more into that. That includes both preclinical and clinical stage biopharmaceutical companies that we're working with. And we're working with the arenas, anybody making plasmid therapies, and there's a large number of companies, believe it or not, making plasmids and selling them. I laugh at my friends, my physicist friends. I can't do it anymore, but I used to just always give them the plasmids. I said, don't pay for those. They're crappy and mine are better. So now, there's a lot of companies making them. DNA vaccines, cell therapies. There are companies, Sangamo is this really nice company out in California doing DNA repair technologies, transfection and targeting agents, electroporation technologies. So these are all people really desperately trying to disguise or make plasmids look like they're smaller just so they can survive the things that I showed you that the minivector already survives. And finally, the people who were developing and trying to, again, make RNAi appropriate for delivery, they're now converting to DNA platforms for longer term effects. And so what we've done is we've executed several non-exclusive license agreements with these companies. We've initiated studies with several more because they want to see it work in their own hands. And it's just been really fun. It's like having a lab times 100, right? Because it used to just be me and one person, and it was me and 10 people. And now it's me and all these companies, right? And they're trying this and they're trying that. It's so amazing to get these data back. Because of course they come and they tell us, and so far, I'm not a salesperson. This is not what I do for a living, but so far, it's all good. I mean, there's no negative thing we've gotten back, so it's really, really exciting. And we're ready to go with the GMP grade mini vectors for phase 1 and 2 trials. All right, so where are we? I realize that some people don't know where Texas is. So here is Texas. It's the biggest in the lower 48 of the states. Specifically, well, I want to highlight something. If you're in New Orleans, and people go to New Orleans seemingly a lot, but they seemingly don't go to Houston as much. But we are neighbors with New Orleans. So when you're in New Orleans, come on down to Houston. And here is the view of the Texas from a Houstonian's perspective. So there is Houston. And just to show you the Med Center and kind of why I took my job, again, I'm a numbers person. So this big data sets, lots and lots of patients is really important to me. And most of my funding is actually on the fluoroquinolone resistance that Dorothy mentioned. And I like to I published a paper with 22,000 patients in my cohort. The next best paper is 11. I mean, there's statistical relevance and there's nothing, right? So I love this. Since I've had my grant, I've had 72 million patient visits in the Texas Medical Center. When people fly in, they think this is downtown, but really you have to look down the street. There is downtown. It's a really remarkable place, and we're very open door. I get the feeling Cambridge is a bit like that now too, because you can go over to this college, go over and talk to other institutions, and the doors are open. So by talking to doctors is how we've made these gene therapy experiments work. And I just want to highlight my lab, which is a wonderful laboratory. Today I talked a lot about the work of Jamie Catanese. He's a staff scientist in the group. Jonathan Fogg, another staff scientist in the group, and Tristan Bodine, who's an MD, PhD, and he's back in medical school now, so we're missing him a little bit. And all my collaborators, and you'll notice most of these are mathematicians and physicists, and they've really made a difference for the research that I've been able to do over the years. And I want to acknowledge my funding, because I have several nice funding agencies that give us money for very basic research that got lucky. And I want to thank the organizers of the meeting last week, and for this particular one, I wanted to thank Keith Moffitt, and thank you for your attention.
Well, this is a bit of a surprise. Perhaps we have one or two comments or questions before we adjourn for tea. Would anyone like to comment on any of the 3 lectures that we've heard? Yes, Dr.
I was
Berich.
just wondering with regard to the brain, Do you have any evidence that your minivectors, if you put them in through the circulation, whether they cross the blood-brain barrier?
So one of my new graduate students is asking that very question right now. But what I do have is I have this fabulous collaborator who's experimenting. She's figured out a way to make a really long needle, and she's doing direct injection into the brain of mice. They go in, they express, they're doing really great things through that way. So we have a way to circumvent that. Now, I don't know how realistic that is for humans, but it seems a little less invasive than drilling a hole in your head. But crossing the blood-brain barrier or the testis blood barrier is also a very important question. We haven't tested that yet, but we're going as quickly as we can.
You can get small peptides through the nasal, and I was wondering whether the nebulizer wouldn't be introducing stuff through the nasal epithelium.
Well, we're certainly going to do that. So we haven't done the histopathology of brain sections yet. We only did the lung because we weren't funded for this, but certainly we've proposed to look everywhere and see where does it spill. Because we may even be able to utilize the lung as an entree into other organs, or the nose as an entree. But at that point, we can start to control it again, mostly by controlling twist and right, believe it or not, right? But that's where we're going.
Yes.
Sorry, I missed the beginning of the talk, but I wanted to know what fraction of cells if you do a transfection, what fraction of cells do your mini circles get into compared to rather larger DNA, normal plasmid DNA?
Well, it's actually a binary response in non-adherent cells and in cells in suspension and non-growing cells. Nothing plasmid-wise gets in at all. And
into which?
Well, we've tried, let's see, what's the name of those? It's a primary cell line. We've done T cells, this is way out of my comfort zone, T cells and dendritic cells and embryonic stem cells, several more that plasmids simply just really struggle to get in at all.
Right.
And in our published work, it was a lymphoma, a primary lymphoma cell line, and a large cell lymphoma cell line, 0 transfection with the plasmid, and it looked like 100% transfection with our minivector.
So every single cell?
Every cell had a minivector in it. So
a minivector that was doing something in it?
We don't know that it was doing something, but if we labeled it with a fluorophore, we could see it in every single cell we looked at. But what we got was complete remission, if you will, of the lymphoma in the mouse. So we don't know, but it was enough to be therapeutic. So we're really excited about that. But if you just look at, HeLa cells, if you try to do transfection, we can say safely 100% with minivector, 40 to 80% with plasmid, but those are very, very easy to work with cells. So those types of cells, there's still a pretty big difference.
Any others?
I wonder, what is the lifetime of these minivectors? What's their size?
Oh, we so want to know. We've only carried out the mouse studies to 7 weeks, and they're at least still expressing at 7 weeks. And maybe that's really good. I don't know how that translates maybe to potential human therapy, and we need to take those out longer. We never dreamed it would go that long, really, because we know that we can find plasmids that have been hanging around in mice for a very long time, but they're no longer expressing. So when we were finding expression after 7 weeks, we got pretty excited. But we haven't found the endpoint yet.
Good, well, thank you again, Lynn. Wonderful talk, a real breakthrough. And thank you to our 2 other speakers, DeWitt and Dorothy. And now we adjourn for tea.